One. Okay, good afternoon, everyone. And we're going to carry on our discussion about evolution, descent with modification. And just to finish off what we were talking about our last class, before we move on to talking about population evolution, I just want to discuss a little bit about the fossil record, because the fossil record is so tremendously important. It features so prominently in any discussion about the evolution, especially of new species and everything. And in Darwin's case, he was fascinated by fossils, and especially once he had made this realization that we could approximate some sort of dating by looking at fossil strata, because fossils that lay at the bottom of a fossil series of sediments were much older than fossils which occurred up at the top. And those simple observations were very informative for him, because he noticed several things about the fossil record. Now, most important is that extinction is the rule of the game. There are far more species which have gone extinct than are currently living. That was his supposition. And he wanted to know, looking at the fossil record, why was it that things went extinct like that? He got his explanation when he realized that there was tremendous intra-specific competition. Members of the same populations compete with one another for resources, and this drives natural selection. And you see, if you follow the fossil record closely enough, you'd see changes as the fossil record proceeded. There's a problem, and that is that things very seldom get fossilized. So there are large gaps in the fossil record. And it can be very difficult to follow a particular morphological story through the fossil record, simply because many members did not get fossilized. So they're just simply not present for us to examine. We are very fortunate in current modern times, because we actually have another kind of fossil record, and that is to be able to go and look at DNA sequence, because DNA sequence preserves ancestry in many ways. We can actually trace ancestry through DNA sequence. And nowadays, we are able to put the two together. We're able to look at the fossil record and often couple it with studies of molecular investigations and have each of them inform one another. So a good example of using a fossil record in this way, we saw before the elephants, for example. Horses are another clear story told by a fossil record. But here is another, and this is the fossil record of the development of cetaceans, of the whales. Now, we know from morphology and now confirmed from molecular studies that the modern day cetaceans, the modern day whales, are actually related to things like the deer, and especially to the hippopotamus, which we have believed for a very long time from morphological evidence, the bone structure and homologies and things like that, that the hippopotamus is in a group with the cetaceans, that the hippopotamus and our living cetaceans share a common ancestor. Extraordinary though it may seem, that is now confirmed by molecular evidence. So looking at what we have living, all we have is the even-toed ungulates, like the deer, and then we have hippopotamus, and then boom, we have the whales. How do we tell the story of the ancestry, of the common ancestry that whales share with animals like the hippopotamus? Well, if we go to the fossil record is going to tell us two very important things. First of all, we look at the fossil record and we find that in the fossil record, there are things which are much closer to the even-toed ungulates than to the cetaceans, but which clearly lie on the same line. And this is an animal here called Pachycetus. But have a look at it. It's fully quadruped. 1, 2, 3, 4. It's got active hind limbs. We believe that it may have been amphibious, may have spent time in water and time out. But Pachycetus is believed to lie in the ancestry of the modern day whales. These forms here, Rhodocetus here, have a look at its hind limbs. Now the limbs are no longer for walking. The limbs now are being adapted for swimming. Those members of this group who swim better are going to survive better. They're going to have more offspring, and their offspring are going to inherit those characteristics. Natural selection is operating to drive the acquisition of alleles, which will modify the hind limbs. Have a look here. The homology is still very great in the hind limbs. They still possess hind limbs, but they have been modified. They are modified for swimming. This organism swam with all 4 limbs. Look at this one here. This is Dorudon. Also, we trace the morphology, and we find now that the pelvis and the hind limbs have been extensively modified. They are no longer used for swimming. And that is like our modern whales. Our modern whales, they lack the hind limb, but the forelimbs are extensively modified for swimming, and so is the tail. So when we look at our modern whales, the cetaceans, we look at the hippopotamus, and we look at this story in between. We believe we can trace a history of the development, a history of the modifications that have taken place. But the fossil record allows us to do something else. And that is, it allows us to put dates on this, because we can actually we can find the strata where these fossils occur, and we can actually date them by various means. I'm not going to go into all of the different ways that we do date, but we can date those geological strata. And we can put approximate dates on the existence of these organisms. And by doing that, we can hypothesize about where the common ancestries were. So the last common ancestor between Dorudon and living cetaceans, about 40 million years ago. Between Rhodocetus and this group here, about 50 million years ago. The common ancestor for all of the cetaceans, we include Pachycetus in the cetaceans, the last common ancestor for all of them, about 67.5 million years ago, something like that. So the fossil record not only allows us to tell the story, it allows us also to begin to put some kind of approximate dating on when these occurred. And surprisingly enough, when we go to the molecular record and do this sort of thing, by and large, the agreement of dates is pretty good between DNA sequence data and of many of the fossil records that we follow.
Hey, Professor, I have a question about
Go
the
ahead.
chart. So basically, with that, what you were explaining with the 5 or 6 animals there is that at one point they all came from like the same ancestors. So the deer and the whale, they kind of have like a connection because they've eventually like mutated into different things and eventually became different
Exactly.
animals. And it's like evolution, is right? If
Correct.
okay,
Correct. What has happened is we've had a common ancestor there for all of them.
okay,
The deer and all of these share a common ancestor way back here, like 62 million years ago or something. There was a population which diverged and gave rise to this line here and then to this whole line here. And so by common ancestor, common ancestor, common ancestor, common ancestor, each of them diverging because of descent with modification.
okay. So each of the spots where like it meets up, that's where like there's a common ancestor that was
Correct.
met.
This
Okay,
this tells you that these guys all had a common ancestor, and it was
okay,
there. Yeah.
okay. Thank you.
Okay. So another tremendous amount of evidence for evolution by the systems of natural selection comes from our studies of biogeography. Now, I've told you before that Darwin, as he traveled around the world, certainly made the observation that there was links, there were animal and plant groups, for example, which occurred on very disparate continents like Australia and South America, say, that were very far apart, and yet there were groups of animals and plants which looked very much like they were closely related. And it was not absolutely clear to Darwin how that relationship, where that common ancestor had been that had given rise to those populations, or how the populations had ended up so far apart. Because Darwin knew nothing about the fact that the shape of the continents and their position on the surface of the earth can actually change. And it does change. 300, 250 million years ago, all of the continents were united into one single giant continent called Pangaea, which then split up. It split first into a huge continent called Gondwanaland, and a northern continent called Laurasia, and those then split into the current disposition of the continents. But when those continents were all together, they were a single landmass. Organisms could move freely across them within bounds of climate type and that sort of thing. But a population that was present on the single continent could actually be separated over time, one from the other. They would preserve many of the characters that they had in common, but each of those populations, once separated like that, could proceed along its own lines of evolution. That is what we now believe. So if we have a look, we can see many examples where there are organisms which have tremendous similarity, separated very, very far from one another. This was an interesting one. This is little freshwater fish. Now, freshwater fish are really interesting because freshwater fish can't make their way across the ocean, right? Freshwater fish in Australia is very isolated from freshwater fish in South America because they have huge oceans that they would have to cross in order for, if that was how they were moving from one to the other. We find that there's this little freshwater fish in a very distinct family called the Galaxidae, and we see them in South America, and we see them in Australia as well. There are 3 species, but there's 2 species here of this family in South America, and there's 1 species here in Australia, but all the way over here in Tasmania. Tasmania, there's islands just south of Australia. And this separation has to have occurred some time ago. These continents are very, very far apart. And we estimate that, in fact, the last common ancestor was present on the giant continent of Gondwanaland about 65 million years ago. Sorry, about 55 million years ago or so. This is just one example, and it's one of many, many, many examples. There are no intermediary species. There's no, it appears clear these must have once shared a common ancestor, and those populations became separate from one another. Here is another one. This is one that's dear to my heart because so much of South African flora is an example of this. This is a group of plants, very, very distinctive group of plants called the proteas. And the proteas are very wide. The family Proteaceae is very widespread. And we find it South America, largely Southern Africa and Australia, and then these species here, which have actually spread up from Australia. Up there. And when we look at them, the different species are absolutely, really radically different to one another, but they share that common feature. They're all members of this family, the Proteaceae. And it's a reflection, again, the proteas were largely, they evolved on the great southern continent of Gondwanaland before Gondwanaland split into Africa, South America, and Australia. These species up here, as I say, this is a little bit different. These have spread up from Australia subsequent to that. So that's just another. There are hundreds of these examples of both plants and animals which share similarities despite huge geographic distance and huge barriers in between them, which would prevent spread, to explain it. So let me just, before we leave Darwin, let me just say one thing. You'll be aware that there are many critics of Darwin's ideas for various reasons, whether often based on belief or whatever, but also very often because people simply can't comprehend the enormity of the time taken for this evolution of modern day organisms to have occurred, etc. And they will often say to you, oh yes, evolution is just a theory. Well, the thing is, a scientific theory is different to the way we talk about a theory in general. In common parlance, a theory is just an idea. It's an idea. It's based on gut feeling or something like that. A scientific theory is not. A scientific theory is an overarching hypothesis to explain something which is so large and so complex, we cannot perform simple experiments to prove or disprove the hypothesis. We have a theory of gravity. Gravity is a theory. We can observe its effects, but there is still tremendous argument for what produces gravity. There's huge arguments about that, but we can observe the effects of gravity. We can do all sorts of experiments about the effects of gravity, and nobody is going to turn to us and say, oh, but gravity is just a theory. It's a real thing. It's just that a scientific theory is too vast, is too great for a simple explanation. And scientific theory very often is broken down into component parts which can be tested, which is exactly what we've seen with the theory of evolution. We can look at things like homology. We can look at molecular homology. We can look at biogeography. These all provide clear explanations of elements of the theory and illuminate it rather. Okay, this is just a final summary of Darwin's ideas about descent with modification. And I emphasize all the way through Darwin's ideas because I want to, before we move on to our next section, I want to make it clear what Darwin did not know. Darwin probably knew virtually nothing, if anything at all, about Mendel's work, because Mendel's work actually was completely obscure right until almost the turn of the century, the 19th and 20th century, when it was rediscovered. So Darwin almost certainly knew nothing about that kind of inheritance or the rules of inheritance. It might have spurred him to many, many new ideas had he done so. They, of course, knew nothing at all about DNA or anything like that. They didn't even really understand the function of chromosomes yet. All of that chromosomal theory, which could have provided Darwin with so much information, was unknown to him. And all of his work was based on observation. He did a huge amount of observation, acquired a huge amount of data, but he knew nothing about genes, and he had no idea that the real mechanism whereby evolution occurs is by changing of alleles. When we see a population evolving, what we are watching happen is the proportion of alleles that are present change. Think of it this way. We just take a homologous pair of chromosomes with two alleles, right? Let's just say that the whole population has just those two alleles for flower color. Everybody's red. Everybody's red. Go there now, count how many alleles for red there are. Well, there is twice as many as there are individuals in the population. But now something changes. There's a mutation, and somebody produces something which is not red. It's not red. And that particular organism is not red. It's a new color and not red. What has really happened? Well, you may well have produced an organism which could be subject to natural selection, positive or negative. But what you've really done is you've changed the number of alleles. Now we've got everybody -2, right? We've got everybody's red except for not red. What you've done is you've changed the proportion of alleles in the population. And that is the stuff of evolution. When we look at populations changing, descent with modification actually means we're watching proportions of alleles change. We're watching the alleles in the population change. That is our next section, and that is the evolution of populations. Because this is really the way in which we unite Darwin's theory of evolution with Mendel and with all of our understanding about genetics, is by looking at how alleles are distributed in a population. We only use the simplest possible models to do this for our purposes, but you'll see exactly what I mean. There's a couple things that this tells us very firmly. When it's a real misconception that people have is that an organism evolves. Well, if you think about it, an organism can't evolve. An organism is as it is. Its phenotype is subject to some impact by the environment. But an organism, a bird with a short, sharp beak, is not going to change through its lifetime and acquire a big, thick beak to eat hard seeds and hand that information onto its progeny. There's no mechanism for that to occur. However, a population of birds can indeed change. A population can evolve. Natural selection operates at the individual. Natural selection decides whether an individual is fitter, produces more offspring, or less fit. And natural selection favors those which are fitter. Natural selection indeed does operate at the individual level, but evolution operates at the population level. We see the population evolve, not an individual. Just to illustrate, there's Darwin's finches on the Galapagos have been intensively investigated for many, many, many decades. They have followed those populations. And fairly recently, in the last couple of decades, there has been a series of really, really appalling droughts that have struck the Galapagos. And when they examine those populations, what they find is that there is a selection for birds which can eat thicker, harder seeds, which are normally ignored. In times of heavy rainfall and everything, there are far more softer seeds and everything. This particular population of finches, they have been measuring the beak size for generations. They had the means, they understood exactly what the beak size was. Now, under normal circumstances, came the drought very, very quickly. From 1976 to 1978, birds with smaller beaks, which only ate small seeds, were strongly selected against. And instead, have a look here, and you'll see that those with a big, thick beak, or the deep beak that could eat hard seeds, were strongly favored by natural selection. So what we observe is a whole population. The whole population changes. But it changes through natural selection, eliminating individuals. If we were really studying this properly, what we could be doing is counting the number of alleles. What we would see is that in the original population of birds, there's tremendous variability. Some of the birds have alleles which would code for big, thick beak. They're probably in the minority. Most of the birds have alleles which code for the smaller, finer beak, because that most of the food that's available, that they're well suited for that. The allele for these beaks is much, much more common. Come the drought, natural selection acts against these, and that the number of those alleles begins to fall in the population. The number of alleles for the thick beak begins to increase. In real terms, when we talk about a population evolving, what we're talking about is a change in the number of alleles, in the proportion of alleles, for one thing or another. And if a proportion of alleles is changing for any reason, evolution is taking place. The population is evolving one way or the other. So we also need to make a distinction between what's referred to as microevolution. Microevolution is this exactly what I'm talking about. It's the change in allele frequencies in a population over generations. And macroevolution, those huge giant changes that take place. So this is macroevolution, is the evolution of the mammals from the reptiles, the evolution of the reptiles from the fish. That's macroevolution. It could also be explained by changes in allele frequencies, but you'd be talking about a huge number of alleles. Very, very difficult to understand. These microevolutionary changes, however, we can explain quite easily theoretically, and we can see physical examples of them in real life as well. Okay, so we're going to now get used to this definition of evolution. Evolution occurs when there is a change in allele frequency in a population over generations. There are three ways in which those allele frequencies can be changed. The first way we've talked about extensively, and that is allele frequency can change by natural selection. That is what happened here. That is what happened here. This population of birds evolved because the allele frequencies changed. The alleles for thick-beak became more common in 1978 than they were in 1976. This population evolved, and it did so by natural selection. Natural selection selected against those with fine beaks and for those with thick beaks. So that natural selection we understand quite well. The next way in which these frequencies change is a process which is called genetic drift. And we will spend a little time talking about this. It's actually quite easy to understand, but it is not a directional process. It's a random process. The other way that alleles can change is by gene flow. And gene flow is when animals with other alleles, with new alleles, other organisms rather, with new alleles migrate in, or when populations are mixed and new alleles are added into the population from another population. That's gene flow. But it's very important. The only one of these which causes adaptation is natural selection. Natural selection is directional only in that it leads to better and better adaptation to the environment. If the environment changes as it did during the drought on the Galapagos, then natural selection operates to select those alleles which are better suited to the new conditions. So only natural selection causes adaptive evolution. These do not. They are more random. Okay, so let's just think, what is it that actually causes genetic variation amongst different organisms? Well, it's caused by differences arising in the genes or in DNA segments. It's changes in the genome of organisms that cause genetic variation. And variability like this is a prerequisite for natural selection. If you haven't got any variability, natural selection has nothing to operate on. And so evolution is not going to take place if there's nothing to operate on. The other thing that is really important is to realize that natural selection operates on the phenotype. It operates on what the organism presents to the environment. That is the interaction between environment and the organism, is through the phenotype. But the characteristics are inherited through the genotype. But the evolution has no way, natural selection has no way of operating directly on the genotype. It has to operate through what is presented by the organism in the phenotype. However, if natural selection operates and it's operating on, it's changing allele frequencies, or the only way it can operate on that variability is if that variability is, the information is contained in the genes, and especially in the genes of the gametes. An organism, if it has a set of alleles which are advantageous, whatever, it's only if its progeny inherit those that natural selection can continue to operate on. Sorry. Is everybody okay? Are you with me so far? If you're there at all. Yes.
Yes.
Are you okay? Are you understanding me so far?
Yes.
Yes.
Okay, all right. Okay, so let me just say. There are various mechanisms whereby new alleles and especially new allele combinations can arise. And one of the most important things is that sexual reproduction, remember back to our discussion about meiosis, sexual reproduction is tremendously important because it actually shuffles genes. It can take genes, from paternal and maternal genes, and it can shuffle them and create new combinations of genes on the chromosomes. And those new combinations then get handed off to the gametes. And we have individual organisms arising with entirely new phenotypes and everything because of those new combinations. But other changes can also take place. The first one will be clear to you, and that is that over time, we accumulate changes in DNA sequence by mutation. And some of those mutations have different consequences, as I'll discuss in a minute. But there's one other thing which you may not know about, and that is that, just think, for example, despite the fact we accept that we all share ultimate common ancestor and that we've shared common ancestries with various things all through evolution, just think about how many chromosomes we have. We have 46 chromosomes, but there are other organisms which have far fewer, and there are some with far more. How is it that the chromosome numbers just are different? We can accept mutation and everything changing DNA sequence, but we must realize that there's actually also changes take place in the structure of the genome, in the nucleus, that there are changes take place in the structure of the chromosomes, which also contribute to evolutionary change. So I'll show you a couple of examples that will make it clear. But remember, for all of these, if the change, a mutation, whatever, is only significant in population terms if it is present in the gametes of an individual and can be handed on to progeny, to the offspring. And the significance of that is that is the only way we contribute alleles to an entire population. The allele, you can think of the alleles as being thrown into a pond, literally into a pool, which we refer to as the gene pool, in the form of the alleles that are present on the gametes. So there are different kinds of mutations, and I think we discussed them when we talked about genes, about the central dogma and everything else, and about genes to proteins and everything. But just to remind you, sometimes mutations have absolutely no effect. They are completely silent. And that is especially the case. Have a look here. These are DNA triplets. There. Here are mRNA codons. And all of these are for leucine, for the amino acid leucine. There are, let's just say we start off with GAA. There you can have 4 different alleles: GAA, GAG, GAT, GAC. 4 different kinds.
Wait, Professor,
Yes.
I have a question. When you say the things that are highlighted in red, this is but is it like adenine, guanine, thymine, or is that
Correct.
completely different? Oh, it is? Oh, okay.
No. Yeah. Sorry. I should have made that clear. I'm sorry. Yes, these are the bases: guanine, adenine, guanine, adenine, guanine, thymine, cytosine.
Okay, I just wanted to make sure.
No, that's fine. No, that's a good clarification. Okay, so the point here is you can have mutations in that last base of this triplet that has absolutely no effect. Look here. They all code for leucine, all of them. Not all the amino acids have 4, like some of them have 2. Methionine, there's only 1 codon for methionine. So it depends. you can actually, there's quite a lot of mutation can occur that remains completely silent. In addition, in the eukaryotes, you can have a lot of mutation takes place in those sections of the gene called the introns, which are never translated to protein. You can have a lot of mutation accumulate there without any phenotypic effect at all. But if a mutation changes an amino acid, if it leads to a new amino acid, a different amino acid being incorporated into the protein that results from translation, very frequently that will be harmful. These proteins have evolved over a very long period of time, and most proteins have evolved to a peak of efficiency. And any change to them actually is usually deleterious. But every now and again, a mutation may occur which improves the function. And any mutation which improves the function, that new allele is going to be favored by natural selection, because it will allow organisms to reproduce more, to exist more efficiently, in fact, reproduce more, and hand on that new mutation to their progeny.
Professor?
Yes, go ahead.
Is it more likely that a harmful mutation will take place rather than a beneficial mutation?
It appears so, yes.
Okay.
Yes. But remember, this is my next point is, remember that those harmful mutations can remain in the population. They are not necessarily quickly eliminated from the population, because they can be hidden as recessive alleles. And then they can remain in the population for a very long time, because as long as the organism has one dominant, then the recessive has no effect, but it remains as an allele in the population to be handed around the gene pool. Now, these mutation rates are very low in animals and plants. On average, it's about one mutation in every 100,000 genes in a generation. That's very, very little. That's very little. There are a lot of the mutations. This is because a lot of mutations are in fact silent, and they're hidden away. Natural selection can't operate on them. Okay, so here's what I meant about some of the chromosomal changes that can take place. There are changes that occur in chromosomes, apparently especially during the process of cell division, and especially during the process of meiosis, producing of the gametes. Chromosomes appear to be able to break. There can be deletions from the chromosomes. They can be disrupted and reorganized. Again, many of these would be harmful, but occasionally it appears that some of them are beneficial. And one of the ways in which these changes can be extremely beneficial is when sections of a gene get duplicated over and over and over and over again. And so what happens when that occurs is that the duplicated sections proceed off on their own line of natural selection. And we have several examples of this. And there's just two that I would mention. The first is in genes associated with detections of odor, smelling. We have 350 copies of a particular gene, and each copy is slightly different and contributes to the suite of smells that we can actually smell, because they produce different products which are involved with reception of smell. 350. We are extremely impoverished because there are others. Mice, for example, have 1,000 of these genes, duplicated smell genes. Dogs have even more. They have thousands. Dogs, it's often said, live in a world of smell. Their other senses, in actual fact, are secondary to their sense of smell is so highly developed. And that is because of this gene duplication. There is another area where gene duplication has been very important, but it's a very complex story, and that is in our immune system. In the genes for our immune system, we see these kind of duplications happening. I just also want to point out to you that chromosome restructuring appears to be very important. And here is just one really interesting example. And this is when we look at the great apes, orangutan, gorilla, chimp, we see that they actually have 48 chromosomes. They have 24 pairs. We have 23 pairs. But when we actually sequence those chromosomes and have a look at them, this is what we see. Here, in the great apes, they are labeled 2A and 2B. So they're different chromosomes. This is 2A, 2B, 2A, 2B, 2A, 2B. But in humans, it appears that an event occurred where A and B, 2A and 2B, were united into a single chromosome, and there it is there. And in actual fact, we can see the great ape segments, if you want to call them that, here, joined into one long, very long chromosome. They have two short ones. We have one very long one. This is not to say that this is what made us human by any means at all. It's just an example that we can see really radical changes in the structure of chromosomes with time. Okay, I'm just sorry, I just need to move something. There we are. So here's another way that we need to sort of think about this, and that is that sexual reproduction, which can shuffle existing alleles into new combinations, especially through crossing over and things like that. Sexual reproduction is also kind of like taking all the available alleles and throwing them into the population. A population is defined as a group of organisms which can reproduce with one another. And it's usually geographically defined. So everybody within that geographic area, with access to one another, they can contribute their gametes. And when they contribute the gametes, what they are doing is they are contributing the alleles that are present on the gametes in whatever particular combinations they are. And the recombination, these recombinations during meiosis are tremendously important because they present new kinds of phenotype to the environment. The phenotype, just think of it in this simple way. Do you look exactly like your father, the men? Women in the class, do you look exactly like your mother? No, no, you don't. You have some similarities to each biological parent, which are by no means identical. Why? Because the genes that ended up contributing to you, the way you look and everything, were reshuffled. You are the result of new combinations of alleles. You're unique. And so what you are presenting to the environment, that is your phenotype. And this shuffling of genes during sexual reproduction allows the development of new phenotypes. And because new phenotypes are result from these combinations, natural selection has new material to work on the whole time. Sexual reproduction is the stuff of evolution. This is how these variants and these combination of variants get presented for natural selection, is through the phenotype. But if we really want to trace the evolution of a population, remembering that evolution applies to populations, not to individuals. If we want to trace what is happening in evolution, what we really want to do is we want to assess the changes that occur in allele frequency. not just in their combinations, but in the actual frequency of alleles present in the population. So again, just some definitions. Population is a localized group of individuals which are capable of interbreeding freely with one another and producing fertile offspring, which themselves can then go on to reproduce with one another. The gene pool consists of all of the alleles for all of the loci in that population. We, in our discussion now, we are only going to trace a few of them, but we need to remember that what we're going to talk about applies to all the genes. In eukaryotes, they are diploid, and all of the cells will have two maternal and paternal copies of chromosomes, and each of those is going to carry alleles for particular genes at particular loci. We say that a locus is fixed in a population if it's incapable of change through sexual reproduction. And the only time that can occur is when everybody has the same gene at the same locus. So everybody has to be homozygous for the same allele for us to refer to a gene as being fixed in a population. We will hear that term. Okay, let's have I just need a sip of water. All right, so we're going to go have a look, and we're going to look at a simple population. And this population of flowers, there are only two alleles for one particular locus.
Wait, Professor, I have a question.
Yes, go ahead.
So, in the pink flower on the bottom, wouldn't that
Correct.
be like? Would that be like the what do you call it? Like the mutation, because it's like two. I'm trying to explain it the proper way. Like it's red and white, so like wouldn't that make, because it's like different? I don't know.
Yeah, I think what you're thinking of is you're thinking back to Mendel. This is incomplete dominance.
Oh, that's okay. Yeah. Incomplete dominance.
Yeah. So, yeah. So what we have here, we only have two alleles. So we have CR, that's red, in the homozygous, that's red. Here is white, CW, and here is heterozygous, CR. Each of them contributing. I'm sorry, this is not incomplete. This is codominance. Each of them is contributing. So this is pink, white. I'm sorry, white, red, pink. How many alleles? How many alleles do we have? Well, the total population of alleles: 1, 2, 3, 4, 5, 6. How many whites do we have? 1, 2, 3. How many red? 1, 2, 3. We're going to start thinking now about populations of alleles. Rather than populations of flowers or whatever, we're going to be thinking about their population of alleles, because there's some strict rules that we're going to encounter about the distribution of the alleles. In everything that we're going to hear are some rules now. First of all, when we're looking at a simple system like this, where there are just two alleles at for one locus, the dominant one we represent by the letter P, and the recessive we represent by the letter Q. And the frequency of all of the alleles for that locus in any population must add up to 1. So P plus Q equals 1. Have a look here. We're going to think about a population with 500 of these flowers. So there's 500 individuals. Each of them has two loci, right? Because two alleles, one maternal, one paternal. We're going to count the alleles. We saw it before. This red is homozygous red. The white, homozygous white. But this one is heterozygous, has one red and one white allele. Let's work out what is the population of alleles. What is the population of alleles in our gene pool? And it's simply done because there are 320, we count, this is the figures they give us. They've counted 320 red flowers, which are homozygous red. So that's how many reds? 320 times 2. But don't forget your heterozygote. Here's the heterozygote, and it also has the red allele there, but it's only got 160 of them. So our total number of reds, 320 times 2 plus 160, 800. Do the same for the white. What have we got? We've got 20 flowers which are white, 20 times 2, because each of them, that's homozygous. And we've got 160 pink. They've got one white allele. So what do we have? We have 20 times 2 there, and we have 160 of these. We've got 200. Add them up, and we've got 1,000. That is correct. We had 1,000 alleles, the frequency of 1. P plus Q equals 1. So have a look here. This is the way we need to think about this actually. Is not in we need to think about it in not in percentage terms, which can get us into trouble, but as a decimal. So what is we said P is the frequency of the dominant, right? And here's the dominant here, that's the red here. We had 800, but we're going to work it out as a proportion of the pole population. It's 800 divided by 1,000. Why 1,000? Why not 500? 1,000 because we're talking about a population of alleles. We're not talking about a population of individuals. We're talking about a population of alleles, and each individual has 2 alleles. The total population of alleles is 1,000. 80% or 0.8. P is 0.8. Q, which is the frequency of the recessive, is this. It was our little sum, 20 times 2 plus 160, 200. 200 out of 1,000, 20%, 0.2. And what was our rule? Our rule was P plus Q equals to 1. 0.8 plus 0.2 is 1. That is correct. 0.8 plus 0.2 is 1. This tells us the frequency that we can simply work out by counting the number of alleles for each. So looking at this, these gentlemen, Mr. Hardy and Mr. Weinberg, devised a formula. And this formula will describe for us the distribution of alleles, in our case for loci, with 2 alleles. And there are certain rules which they want us to obey. First of all, when individuals in the population mate, they do so completely freely. There's no barriers. In other words, no one person is selecting a mate in a way which could influence the distribution of alleles. Mating is effectively completely random. Second of all, the inheritance is simple, is Mendelian. It obeys the Mendelian inheritance. We're not going to be talking about perhaps things like with multi-genic contributions and things like that. It's simple Mendelian genetics. The next is that the allele and genotype frequencies remain constant from generation to generation. Well, what would be changing them? Well, one of the things which could be changing them would be mutation. Right? So there's no mutation taking place. there's no immigration or emigration, which could bring in new alleles. Now, as long as these conditions pertain, they maintain that the population, the distribution of alleles remains constant. And that population is said to be in Hardy-Weinberg equilibrium. If the equilibrium is disturbed in any way, evolution is taking place for one reason or another. The only way in which the distribution of alleles can be pushed out of Hardy-Weinberg equilibrium is if evolution is taking place. So this gives us a very useful little model. We can look at a population and see, does it obey Hardy-Weinberg laws? Is it in Hardy-Weinberg equilibrium? If it is, no evolution is taking place. If it isn't, something is happening, and that something is evolution for one reason or another. And remember, I gave you 3 reasons why allele frequencies could change: natural selection, gene flow, and genetic drift, all of which we will talk about in a while. So there is an equation. It's called the Hardy-Weinberg equation, and it describes the genetic makeup for any population which is evolutionarily stable for a particular locus. No evolution is taking place for that particular locus. Maybe taking place at other loci, but for the one that we are considering, there's nothing happening. It's remaining in Hardy-Weinberg equilibrium. And if it doesn't obey those, that's very interesting. Then we will start to look at that population at that locus very carefully. Why is it not obeying Hardy-Weinberg? What is happening to push it one way or the other? Here are some useful ways to think. I know that this is confusing and not necessarily that easy to conceive of. But here are some different ways of looking at what I'm trying to tell you. And emphasizing again, what we are talking about is not a population of individuals. We are talking about populations of alleles. That's the way we need to think about it. Here are our flowers. Okay, this is our flower population. And now we've done away with the flower and we're simply looking at the alleles themselves. And we're going to throw them into a gene pool. And here is the gene pool. It contains all of the alleles in that population. And we saw what they were. We saw what the frequency was. The dominant allele, the CR, was 0.8. And that was, we have here 20 altogether. So that's 16 out of the 20 alleles in the gene pool here. Those are the dominant. And 0.24 out of the 20 are the recessive allele. Now, here's the thing, is that when we produce eggs and when we produce sperm, it is effectively a random process. The alleles are picked at random. Why do I say that? We'll go back to meiosis and remember that the whole chromosomes are separated from one another during the process of meiosis, and that separation takes place at random. And what you realize is that for a large population of eggs, a large population of sperm, that because the selection of alleles is at random, it's going to reflect the same proportions. So when each egg, for example, there's an 80% chance that it will have the dominant allele, 20% chance it will have the recessive. Same for the sperm. 80% chance it will have the dominant, 20% chance that it will have the recessive. And when we now put them together, well, we're going to see something very, very interesting. Remember that the first thing, get it right into your head, and because every problem that you ever see with this is easily solved once you remember this. P plus Q equals 1. Okay, the frequency of the alleles always adds up to 1. And if we go now to our same population of wildflowers, we saw that it had 1,000 alleles. If you don't remember, just flip back a couple of slides and you'll see there were 1,000 alleles. Now what we're going to do, we're going to take eggs and we're going to take sperm. And we remember we've got a large number. So we've got a large number of eggs, a large number of sperm. So it's going to reflect those same proportions. Here are the sperm. Dominant, 0.8 of the sperm. Recessive, 20%, 0.2 of the sperm. Here, dominant, recessive. Now, what we're going to do is we're going to cross them. So we got 0.8 times 0.8, 0.64. That is p squared. We look at the recessive. We've got here 0.2 by 0.2 to produce the homozygous recessive. 0.2 by 0.2, 0.04. That is q squared. And the heterozygotes, PQ, P 0.8 times 0.2, 0.16. Same here. CW, CR 0.8 by 0.2, 0.16, 2 PQ. This is the Hardy-Weinberg formula. This is the Hardy-Weinberg equation, rather. The Hardy-Weinberg equation states P for a population in Hardy-Weinberg equilibrium, these proportions are preserved. That when we look at a locus with 2 alleles, this will be the distribution. It will be given by the formula p squared plus 2pq plus q squared equals 1. And here, this is how we can, if you forget how we derived it, just come back here and do this Punnett square over again and do these multiplications, and you'll see how the Hardy-Weinberg equation is actually derived. P squared plus 2pq. There are 2 ways in which you get the heterozygotes, plus q squared equals 1. And by looking at this, what we're going to realize is that we can, in fact, derive any part of that equation once we know some of the features of the distribution of the alleles. So here, for example, the frequency of the genotypes is calculated. We said p squared, 0.64. The frequency of the heterozygotes for that population, 0.32. The population of homozygous recessives, 0.04. And all of them, add them all up, and it will all add up to 1. So this formula, p squared plus 2pq plus q squared, describes a population which is stable. No matter what you do, that the alleles in that population are going to always sort in exactly the same way. And as long as that is the case, then nothing is happening with this population. There's no evolution taking place. And you can, in fact, using this formula, I can begin to work out some important things about populations that we're looking at, as long as I assume that there's no reproduction taking place. I mean, no evolution taking place. So it's obvious that this is a theoretical approach. In real populations, change does take place for all sorts of reasons. Mating, for a start, is seldom entirely random. Populations are seldom entirely isolated, and there are various other things that can take place. Nonetheless, it remains a useful formula, as I'll show you when we do a few problems. So these are the 5 conditions for a Hardy-Weinberg population which remains in equilibrium for non-evolving population. No mutations taking place. Mating is completely random. There's no sexual selection at all. There's no natural selection taking place. The environment and everything is stable. We need a very large population size for reasons that will become clear in a while when we talk about genetic drift. And lastly, there's no gene flow. We've got no new alleles coming in. We've got no loss of alleles by emigration. Instead, the population remains stable as far as gene flow is concerned. Let's do a real example. We're going to look at a gene for phenylketonuria. Phenylketonuria is a metabolic disorder. It can, in fact, be a fatal disorder, but it can be avoided if an infant is put onto a special diet. It's a failure to metabolize an amino acid properly, and it results in neurological damage. Now, we can look at the whole population of the United States. And first of all, the gene mutation rate is very, very low for PKU. So we don't have new PKU alleles arising. That's the point. We have a certain number of PKU alleles present in the population, and it remains relatively stable. So people are not selecting one another in general as to whether they do or do not have the PKU allele. They just don't know, by and large. And so there's no selection. There's no sexual selection for that. There's sexual selection for other things, but not for that allele. Now, the natural selection will occasionally operate, but it will only operate on homozygotes who have 2 copies of the PKU allele. That's when the disease makes its appearance, is in the homozygote who has 2 recessives, because it's a recessive allele. So that would be very, very, very rare. And in fact, has almost no impact on the size of the PKU allele population in the whole population. The population is very large compared to those with the allele. And if we look at other human populations all around the world, they have similar allele frequencies for PKU. So emigration is not going to have much effect. Within certain bounds, this population, these alleles obey the rules for examination for Hardy-Weinberg equilibrium. We can suggest that it is probably in Hardy-Weinberg equilibrium. So now, how do we use it? Well, let's ask this question. The appearance of somebody with phenylketonuria it's quite easy to identify them because there are certain metabolic markers that make them immediately apparent. So that's fine. We can treat them and everything. But what we really are interested in is how many of these alleles are present in the population overall. How many PKU alleles are present in the American population total? The Hardy-Weinberg equation can tell us. First of all, we can see that the occurrence of phenylketonuria is about 1 in 10,000. Phenylketonuria only appears in the homozygote. So Q squared here, Q squared is 0.0001. Q squared is 0.0001. So if we did a Punnett square, right? And we ended up with that down in that lower corner, we ended up Q q squared. And this is the frequency 0.0001. So q here is equal to 0.01. If q is equal to 0.01, what is p? Well, p plus q is 1. So 1 minus 0.01 gives us p. P is 0.99. Now it's a simple matter to work out how many heterozygotes are there in the population. And here they are here. 2pq is 2 times 0.99 times 0.01. 0.0198. Approximately 2% of the population are carriers of the PKU allele. And we worked it all out simply because we knew how many PKU, how many births there were that result in phenylketonuria. So that is the way in which you would use this. Okay, so I'm stopping here. It's about a few minutes early for the class, but I want you please to go to these 2 YouTube videos. And both of these YouTube videos discuss the Hardy-Weinberg equations and how to solve Hardy-Weinberg problems. And explains it very simply, very clearly. Sit with a pen and paper and follow through. And then we'll try it out on Wednesday, okay? And on Wednesday, we'll have a quiz. And I'll try and design some good Hardy-Weinberg problems for you to see how well you do. Okay? Are there any questions?
Yes, Professor, how many questions will the quiz have?
Oh, I don't know, 5?
Okay. Do you know how much time we will have?
I beg your pardon?
Do you know how much time we will have?
5. You'll be working things out. So I would say probably 15 minutes, 20 minutes.
Okay.
5 minutes a problem, something like that. And I'll watch and see what's happening. And if it looks like people are getting hung up, then I'll give you a little bit of extra time. Anyone else?
Yes. So after we take the quiz, are we going to be able to see what we got wrong?
Yes. Now, Edward, you should be able to see
Yeah,
all the.
I did. Last time I was making a checklist, I was looking at what I got wrong, and I was trying to understand why I got it wrong. So yeah, I did see it.
Okay. All right then. And I'll do the same thing. I'll do the same thing with this. All right.
Thank you, Professor.
All right
Thank
then.
you so much, Professor.
Good afternoon, everybody, and I'll see you again soon.
Professor, I have a question about our grades for those assignments.
For assignments, yes. Go
Yes.
ahead.
When will those grades go into play with our overall grade for the class?
Are they not being posted already?
And well, on the separate MyLab account, it is posted. Like I can see what I got after I do the
Yes.
assignment. But does that grade go onto Canvas?
Yes, it should be on Canvas.
Okay, because the last time I checked, I didn't see any of those. I just saw it on the
All right.
separate page.
I'll check up again. But yeah, as long as it is there on the assignments, then if it's not appearing on Canvas, it's a simple matter of some bookkeeping
Okay.
to get it to appear. So I'll check up on that for you.
All right, thank you.
Okay. All right then. Okay, everybody. Then I'll see you again next time.
I'll see you next Wednesday. Bye, Professor.
Professor.
Bye bye.
Thank you.